Hello, I'm Maria Williams with the Collaborative on Health and the Environment. CHE enjoys bringing you the latest environmental health science through our partnership calls and webinars, science serves, publications, and social media. I would like to welcome everyone to today's CHE partnership webinar, which is titled, Using Molecular Epidemiology to Understand Chemical Threats to Early Life Children's Health. Our moderator today is Sarah Howard, founder and manager of diabetesandenvironment.org. We will leave time following the presentations for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentations. After the presentations, our moderator will read out questions for our panelists to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speakers. This webinar is scheduled to last for 45 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Sarah.
Thanks, Maria. Hello, everyone, and welcome. Our speakers today are Allan Just and Joe Braun. Allan Just will go first. He is assistant professor in the Department of Environmental Medicine and Public Health at the Icahn School of Medicine at Mount Sinai. His research interests include children's environmental health, computational methods for epigenomics, endocrine disrupting compounds, and modeling air pollution using remote sensing. He uses large data sets, computational tools, and molecular epidemiology to investigate the links between environmental exposures and children's health. The second speaker will be Joe Braun, who is assistant professor at the Department of Epidemiology at Braun University School of Public Health. He is interested in the health consequences of early life environmental chemical exposures in pregnant women, infants, and children, especially things like obesity and neurodevelopmental disorders like ADHD and autism. He's also interested in identifying modifiable sources of environmental chemical exposures in pregnant women and children. So with that, Allan, would you like to start?
Sure, thanks, Sarah. So I'm going to talk today a little bit about epigenetics as a biomarker in environmental health. And often in our environmental health paradigm, we're thinking about relating some sort of exposure, whether it's a chemical or a non-chemical stressor, to health outcomes, and some important health outcomes were just listed. And so people have been getting excited about the epigenome, or a set of biologic mechanisms that play a role in transcriptional regulation, as a potential mediator in between, and this has gotten people very excited both in the developmental origins of health and disease, but more generally in the environmental health community. And I'm going to talk about why the epigenome might be useful as a biomarker, and also some of the challenges and difficulties with these new approaches enabled by new biotechnology. So I'm going to speak mostly about DNA methylation, which is a modification to the DNA. It's a chemical chemical change where a methyl group is added to the exterior of your DNA. It's very well studied because it often can decrease gene expression, but it's also quite stable, and we have very good tools to assay DNA methylation. So originally, and this is kind of commonly the case in new fields, people were looking at specific hypotheses where they were testing the relationship between methylation and exposures, or methylation and outcomes, looking at just one gene or a few genes that are known as candidate gene approaches. But now we have new tools, epigenome-wide platforms, and there's this very efficient set of techniques that use DNA microarrays. So your DNA that you're assaying, it will hybridize to that. I have here one of these arrays with me that would assay 12 samples, and each of those 12 samples you would measure almost 500,000 sites, but now we do more than 800,000 sites, distinct DNA methylation sites that you would look at in each sample. And so this is really opened up the ability, the scope, and so instead of looking at a single candidate gene, now people are conducting what are known as epigenome-wide association studies, simultaneously considering hundreds of thousands of candidates where methylation sites might be associated with exposure or with outcome. Now, an environmental example that we look at how early exposures leave a fingerprint on the epigenome is looking at smoking. So a number of studies have been done with smoking, and this is some results from a meta-analysis that I participated in, came out last year from a consortia that I'll introduce later. But these 13 cohorts brought together all of their data from more than 6,000 newborns, and they compared the DNA methylation of newborns born to women who had smoked throughout pregnancy. They compared those to the DNA methylation of newborns of moms who had not smoked, and then they adjusted for a number of important covariates. And what they found was not only were there hundreds of these methylation sites that seemed to have very strong statistical signals of being associated with that early exposure to tobacco, but that it was really a signature. They were coming in characteristic locations. So what I'm showing here, the x-axis is the organization of your genome. This is known as a Manhattan plot. And so these are all of your chromosomes sort of just lined up. And the y-axis is the strength of the statistical association, where points that are higher up are stronger, more strongly associated. And this is after doing all those hundreds of thousands of tests. Some of these points are incredibly strongly associated, and there's an organization to them. So there's a number of sites that are near each other in the genome that all share an association with this tobacco smoking, forming something of an interpretable signature of the regions in the genome in which the methylation is altered in association with that early exposure. And we can start to delve into trying to understand what are the gene regulatory consequences of the altered methylation in those locations, right? So it's very specific. And we find different specific signatures, different locations in which we have these associations when we look at different kinds of environmental health exposures, although it can be shared. So for example, some of the same signatures that we see in babies that are born to women who smoke during pregnancy, we see in adults who smoke or who used to smoke. So there's a commonality to that signature. One of the challenges that's facing these this new field that makes it a little bit maybe different from genomics, where you're studying the gene sequence, is that the epigenome, or the DNA methylation that I'm going to talk about, it's different in every cell in your body, and it's particularly very different between tissues. So we need to think about the disease process that we're interested in, or the exposure that we want to get a biomarker for, and think about what are the tissues that agent may be acting on, or that are etiologically relevant. Also, we know that often we get samples that are a composite of many different kinds of cells. So blood is made up of many different kinds of cells, the DNA in your blood, and each of those cell types has characteristic methylation. We have to be careful that we're distinguishing differences in methylation and differences in the composition of the cells that make up the samples that we collect. But I'm also going to talk today about some of the analytic challenges that I alluded to because of the high dimensionality of the data, that we're looking at so many different things. We're doing so many statistical tests at the same time. And this creates something of a higher bar where we have to be both careful when we do our analyses, but it's also more difficult to dive into them because there's so many different components to our data to look at simultaneously. So because other fields have faced this before, but I think that this is something of a new challenge for environmental health in the modern era, that we're now swimming in data, and this creates a need for new diagnostic tools, and we have to be able to check the same assumptions and results that would apply if we were looking at a single exposure and outcome, but now we have to be able to scale that up. Because it's possible to look at so many measurements simultaneously. And these fluorescence-based microarrays, they're quite complicated. There's a number of steps that are necessary to come up with the kind of empirical data that we use in our epidemiologic epigenetic studies. And so there's a complexity to that needs to be respected. One of the things that I've been working on recently is sort of going back to basics, thinking about quality control, which is important for any kinds of science. But here, I think it's particularly important because of the complexity of the data. So we're coming out with new pipelines for quality control, even those earliest stages where you can ask yourself, did this work? You know, not just do I have measurements from every one of my samples, but are those measurements coherent, consistent? Is there evidence of some sort of failure, mix-up, or contamination? We have an R package developed primarily by my postdoc, Dr. Jonathan Heiss, called EWAS tools. So EWAS is Epigenome-Wide Association Study. We're coming up with tools to help groups that are undertaking those sorts of studies. And we have a new bioarchive preprint that we put up about how do you trust that the measures that you've gotten are actual signals and not just coming from the noise, and that this needs to be automated, but also needs to be done, you know, in a thoughtful, empirically driven fashion, using publicly available data sets to sort of check these assumptions. We know that even low numbers of misidentified samples or very large outliers, which can occur in these data sets, they can really mess up our ability to come to interpretable and robust findings, particularly because we have to be fairly stringent about calling something as a real association because we're looking at hundreds of thousands of methylation sites simultaneously in the case of these epigenome-wide association studies. We've done some studies where we use technical replicates, samples that we run the same exact sample more than once to quantify our uncertainty. So if you run the same exact sample twice, then any differences that you see between those measurements, that has to be noise, right? That's technical measurement error. And we can use statistical techniques to quantify that. And we see that the sites that have more technical error, either because they don't vary a lot in the general population, or because we weren't able to do as good a job at measuring those sites maybe need to be discarded because we're not going to find our robust and rigorous results in those sites. So, you know, there's a whole suite of different analytic approaches that we're investigating now, thinking about how do we help the signals that we're interested in, these epigenomic signatures, how do we help these rise to the top? And so I mentioned sort of prioritizing features in our measurement, in our data sets that have been measured with more accuracy or precision. Also thinking about prioritizing features that are more biologically relevant. So a lot of these epigenome-wide association studies that we've done, whether it's related to tobacco or alcohol or air pollution, we're seeing an emerging pattern that many of the methylation sites that show up as associated with environmental exposures, they're occurring in a particular location in the genome, these kinds of functional elements known as enhancers that play a role in transcription factor binding, and that might be why they're relevant to the regulation of gene transcription. And so, you know, maybe we want to prioritize or think about why those enhancers might be where we want to focus our energies as we are scanning across all of these potential hypotheses. Similarly, we can borrow information and say that methylation sites that are in genes that are related to each other biologically, they can be organized into pathways, and we can try to use that information to inform and improve our analytic approach. Or we can look for methylation sites that are near each other, just as I showed that pattern with the tobacco smoking, where some of the strongest results were from very distinct regions in the genome where it almost looked like those points were stacked on top of each other because there are many methylation sites that are near each other. We can think about regional analyses or methods to look for coherence, where the methylation in one particular region is associated with our outcome or our exposure of interest. So you know, one of the ways that we're addressing this challenge, where, you know, looking for everything makes it a little bit harder to find anything, is that we're teaming up. So there's a couple consortia where people are using meta-analytic approaches, bringing together multiple data sets. One of the consortia that I work with quite a lot is the Prenatal and Childhood Epigenetics Consortia, or PACE. We recently had a cohort profile that was published in the International Journal of Epidemiology. I encourage you to look at that if you're interested in finding out more about pace. And also there's a very well-established consortia called the Cohorts for Heart and Aging Research in Genetic Epidemiology Charge, which has an epigenetic working group that's doing some amazing work, primarily focused in adults, looking at epigenetics in relation to disease outcomes primarily. So this is another strategy by teaming up, by harmonizing data sets and bringing them together into meta-analyses, pooling our information. We have more statistical power, even when we want to look at these broader data sets. And that can help when we think that the challenge is that we don't have enough statistical power, that there's just too many things that we're looking at, and it's hard to exclude chance. But that's not the only problem that we might encounter. And so we can't just be careful about how many tests we're doing. We still have the epidemiologic conundrum of facing down confounding and being careful about that, which is even harder maybe in the high dimensional setting where there's just so many different things that we're looking at simultaneously. And so I think it's important, even as we branch environmental health into these new eras using higher dimensional data, that we can't forget the assumptions and the pitfalls that we've always had to deal with, particularly in observational studies. So in summary, I think that epigenomics is a very promising and emerging area for environmental health. I gave an example with early environmental tobacco smoking or maternal exposure to the fetus, but Joe's going to give an example in his talk, another environmental exposure. And so I think that this could be very promising because of the specificity of the signatures and the potential biologic mechanisms that link these signatures with gene expression. And the measurements are standardized. That's allowed the field to blossom because it's possible to very efficiently make so many epigenetic measurements, but it's now the analyses that are complicated, right? And the high dimensionality is part of that challenge. And so we need care, particularly when our sample size is much less than the number of things that we want to look at. So maybe we can borrow information using biological prioritization or statistical techniques. So with that, I thank you, and I turn it over to my co-presenter.
Thank you, Alan. And we're going to do questions at the end. So save your questions. And Joe, are you ready?
Yes, I am. Can you hear me?
Yep.
Okay, I'm going to share my screen. All right. Good afternoon, everyone. I want to thank the Collaborative for Health on the Environment for inviting me here to talk to you today. I'm going to talk about some of the links between human perfluoroalkyl substance exposure, childhood obesity, and some of the underlying molecular mechanisms that we've been investigating. So perfluoroalkyl substances, or PFAS, these are a class of persistent environmental chemicals that are used in a wide variety of commercial and industrial products. They are used in stain and water repellent coatings. So for instance, in products like Scotchgard or in some food packaging materials to keep oil and water from mixing. They're also used in cleaning agents, cosmetics, used in aqueous film forming foams, which are used as firefighting foams on Air Force bases to stop fires. And they're also used as industrial surfactants to make other fluorotelomeres and fluoropolymers. There are thousands of these chemicals. We predominantly study some of the shorter chain molecules like PFOA and PFOS that you see up there in the upper right-hand corner, which are these have these long carbon chains with a fluorinated backbone. And it's these fluorinated backbones that make these chemicals so persistent in the environment, because the carbon fluoride bond is the strongest organic chemistry bond there is. And there's very little environmental, chemical, or biological degradation of these molecules. Now, some of these chemicals have been phased out of use here in the United States, PFOA and PFOS specifically. This is done voluntarily by industry, but there are concerns over the replacements as well as the thousands of other perfluoroalkyl substances that are used in commerce and industry. In terms of human exposure, as I mentioned, these are persistent chemicals and they have biological half-lives on the order of 3 to 7 years. And in some cases, in the environment, they can hang around for decades or more. And in some cases, maybe even longer than that. Many of these perfluoroalkyl acids are detected routinely in human serum. So PFOA and PFOS, which I showed on the prior slide, are detected in nearly everyone in the United States, as well as in other Western populations, as well as China. And we think that diet is the predominant source of exposure for most adults. Now, there are some exceptions to that. For breastfed infants, breast milk is probably the biggest source of exposure, and infants who are breastfed actually have 14 to 15 times the amount of exposure to perfluoroalkyl substances as do non-breastfed infants and adults. And we also know that in some communities there's been drinking water contamination with perfluoroalkyl substances that's resulted in very high water exposures that could rival those that are observed or exceed those that we would have from diet. So there are some exceptions to the rule that it's usually diet. Now there's a wide array of health concerns that we are concerned about with regard to perfluoroalkyl substances, and there's varying degrees of certainty about their causality when we look at the epidemiological and animal studies. So the National Toxicology Program recently released a report that declared that PFOA and PFOS are immunotoxic in both animals and humans, and there was quite a bit of evidence of that. There's some evidence that these substances are associated with some cancers. Less evidence, but some suggesting that these substances affect neurodevelopment and could affect the risk of some behavioral disorders. They also have effects on the liver and increasing the risk of fatty liver disease. They can affect thyroid function, as well as cholesterol and triglyceride metabolism. And there's also general concern about how they affect fetal, neonatal, infant, and child growth and development. And this is with special concern for these vulnerable populations because we know the fetus and infant and child could have higher exposure to these chemicals, as well as immature detoxification pathways to excrete them. So in terms of some of the health effects of these chemicals in infants and children, the perfluoroalkyl substances can affect several different biological mechanisms that are important for growth and metabolism. These chemicals, PFOA and PFOS, specifically can activate the peroxisome proliferator activated receptor alpha and gamma. These are two receptors that are important for lipolysis and adipogenesis, respectively. They also can inhibit an enzyme called 11-beta hydroxysteroid dehydrogenase number 2, which I abbreviate as 11-beta HSD2 there. This is an enzyme that's involved in cortisol metabolism, and it's actually a very important enzyme for us as adults as well as for the fetus in that it inactivates cortisol and prevents excess cortisol from accumulating in our bodies. And in the fetus, this is important as well because you don't want the fetus to have too much cortisol. But these chemicals, PFOA, PFOS, and some of the others, can inhibit this enzyme. And in some animal studies and in vitro systems, the inhibition can cause increases in circulating levels of cortisol. The perfluoroalkyl substances also have been associated with lipid and triglyceride metabolism in humans, specifically causing increases in HDL, LDL, and triglyceride concentrations in humans. But there's different effects in animals than there are in humans, so there are some species differences in terms of what these chemicals can do. So these concerns about how these substances affect these biological systems that are important for growth and metabolism have led to concern that perfluoroalkyl substances are obesogens, or chemicals that could increase our risk of obesity or associated cardiometabolic disorders. And there are some studies showing that PFOA and PFOS are associated with fetal growth, specifically reduced fetal growth. Two meta-analyses conducted by Tracy Woodruff's group out of UCSF looked at animal and human studies that had looked at PFOA in relationship to infant birth weight, or in the case of rodents, they were the pups. And what they found was remarkably consistent evidence in both animals and humans showing that PFOA exposure was associated with decreases in birth weight. Specifically, each nanogram per milliliter increase in PFOA was associated with about a 19-gram decrease in birth weight. And then there was in the rodents, about a milligram per kilogram per day increase in PFOA was associated with about a 23-gram decrease in the pup birth weight. And this is amazing to sort of see this consistency between animal and human studies, and even within the human studies, which I'm showing a slide from the paper here. This is the forest plot of the different studies where each individual study's point estimate is shown, along with a 95% confidence interval for that estimate, and then the size of the box indicates the size of the study. And what you see is that almost all of the studies, with one exception, had an inverse association between PFOA and neonatal birth weight. Now, the potential effects of PFOA on fetal growth have raised concern about how they might affect offspring growth. And in several studies, both prenatal PFOA and PFOS has been associated with increases in adiposity as well as altered growth trajectories. A couple of notable studies are one from Sharon Sagiev's group using Project Viva, showing that PFOA and PFOS exposures were associated with increased adiposity at 8 years of age, but not at 3 years of age. And this was in a cohort of about 700 children or mother-child pairs. And in another study out of Denmark, their prenatal PFOA levels were associated with increased adiposity measurements, specifically BMI and waist circumference, in 20-year-old females, but not males. And this sample size, this study had about 600 participants in it. We previously showed in a cohort from Cincinnati, Ohio, that prenatal PFOA exposures were associated with increased adiposity at age 8 years of age, as well as alterations in growth trajectories in about 285 children. And this slide here on the right is showing you those growth trajectories with respect to prenatal PFOA concentrations. And what you see is that children, what was plotted here was the mean BMI Z-score between 2 and 8 years of age among children in that cohort. And the solid black line there is the BMI Z-score trajectory for children in the lowest PFOA tercile, and they gained about 0.1 BMI Z-score units between 2 and 8. The children in the second tercile are the dashed line, and they had a BMI that was similar to children at 2 years of age, in the first tercile at 2 years of age, but then by age 8, they had gained over 0.4 BMI units relative to their baseline. And children in that third PFOA tercile, who are the dashed dotted line, had a growth trajectory that was different in that they started with a much lower BMI at 2 years of age compared to children in the first and second tercile, but by age 8, they had gained more BMI than children in the first tercile, such that they were almost the same body mass index. And what's important to note here is that the exposure here, PFOA, was associated with not only differences in the absolute level of adiposity at any age, but also the way that children grew between 2 and 8 years of age. So we need to think not just about how a chemical can affect you at any given time point, but how it affects you over time. There have been some studies investigating how perfluoroalkyl substances affect breastfeeding. There have been 3 studies that I'm aware of showing that both prenatal PFOA and PFOS concentrations are associated with decreased duration of breastfeeding. And we know from animal studies and in vitro work that perfluoroalkyl substances can influence breast development, as well as the secretion and action of lactation hormones. And this is a potential mechanism that explains the association between PFAS and obesity, because we know that breastfed infants tend to not gain as much weight as bottle-fed infants, and that breastfed babies tend to be at decreased risk of being overweight or obese later in life. Now, continuing with the theme of the molecular epidemiologic tools, we've been doing some work looking at perfluoroalkyl substances and epigenetics. And Allan gave a great introduction to epigenetics, so I'm not going to rehash any of that here. But there have been some studies that have begun looking at perfluoroalkyl substances and epigenetics. Deb Watkins found that PFAS exposure was associated with higher line 1 methylation in a cohort of highly exposed individuals from the West Virginia area. There have been some other smaller studies showing that PFOA was associated with global DNA hypomethylation, and a study finding that changes in DNA methylation of growth-related genes in a cohort from Japan. And also from the same cohort in West Virginia, PFOA and PFAS exposure were associated with expression of cholesterol genes. In this case, they didn't look at DNA methylation, but they looked at RNA expression in the blood. Now, these studies have been more targeted in nature, where they've looked at proxies for global DNA methylation or methylation of specific genes like insulin-related growth factor or cholesterol genes, but no one's taken an unbiased or untargeted approach to this to see, are there potential associations with pathways we might not know about. So we did this in the home study. This is a prospective pregnancy and birth cohort study from Cincinnati, Ohio, where we looked at 44 mothers and children, and selected those with the lowest PFOA levels, so 22 mother-child pairs with the lowest PFOA levels, and then the 22 mother-infant pairs with the highest PFOA levels during pregnancy. So we tried to ensure that we had a large exposure contrast. And then in the infants, we measured leukocyte DNA methylation in the cord blood using the Illumina 450K array. As Allan mentioned previously, there's concerns about cell type and the data processing, and so we did account for many of these factors in our analysis, specifically accounting for cell type. And this is one of the results from that study. This was done by Samantha Kingsley, who is a postdoc working with me, and she did this while she was a graduate student at Braun. And what we found was that PFOA was associated with hypomethylation in many cases of lots of genes, and there was an excess of these associations in the promoter regions of many genes. Among our top 20 hits, none of which were statistically significant at a false discovery rate correction p-value of 0.05, we did find that there were 7 CpG sites that were consistently hypomethylated in 3 different genes. And we found some notable genes that were associated with PFOA exposure when we're considering the potential developmental toxicity of PFOA. Specifically, we found hypomethylation in the RASA3 gene. RASA3 is an inhibitor of RAS, the RAS protein, which is involved in cell growth and differentiation. So this may explain some of the growth restrictive effects that PFOA has. We also found that PFOA exposure was associated with hypomethylation of the OPRD1 or the opioid receptor gene. And this gene, or polymorphisms in this gene, have been associated with obesity in adolescents and adults. And we also found hypomethylation at one site in the HOXD3 gene. HOXD3 is a homeobox gene, and these are a family of genes that are conserved across multiple species and are involved in embryogenesis and the programming of positional axes in the body, specifically how to tell your body which side is left and which side is right. We also found that these top 20 CpG sites that we looked at were perfectly predictive of the PFOA exposure group. And as I noted before, we had 22 in each group, and we were able to use these top 20 genes to perfectly predict that when we used a cluster analysis. We're doing new work in this cohort to measure leukocyte DNA methylation in over 200 children at delivery, as well as at age 8 and 12 years of age, and then relate PFOA as well as other perfluoroalkyl substance exposures to leukocyte DNA methylation, both in the cord blood, and then to see if these associations persist as children age. I want to close and briefly talk about perfluoroalkyl substances in drinking water, because this has been a very important topic lately that has made the news. Many of you have probably seen headlines about the Wolverine site in Michigan, which is an old tannery site that was using perfluoroalkyl substances and had been dumping them, and they resulted in groundwater contamination. In my home state of Rhode Island, over 200 Burrillville residents have contamination of their drinking water with perfluoro substances. And there have been many other notable high-profile cases of drinking water being contaminated by these perfluoroalkyl substances. In May of 2016, the Environmental Protection Agency established a drinking water health advisory level for PFOA and PFOS. And specifically, this level is 70 nanograms per milliliter for PFOA, PFOS, or their sum. So what's notable here is that it considered the cumulative exposure to both compounds. And this threshold was established based on rodent studies, and in PFOA, it was for decreases in bone formation and accelerated pubertal development. And for PFOS, it was because of associations between PFOS and offspring weight. It did account for the susceptibility of the fetus and infant, so there was an additional uncertainty factor incorporated in there. But it's important to note that this is a non-enforceable and non-regulatory level. This level isn't going to be reported and acted upon to protect people's health unless it's done at the state or local level. Now, there's some tools available for people to use to incorporate measurements that they may have. People may be going out and getting measurements of these perfluoroalkyl substances in their water as well as their serum. And Scott Bartell at UC Irvine has been doing some work to make this information more accessible. And he's developed a web-based calculator that allows you to input your measured PFOA serum levels as well as water levels that you might have in your community. And what he can tell you is what your PFOA levels would be after ceasing water exposure, or what your PFOA levels would be after you were being exposed to water that was contaminated with PFOA. This calculator is based off of a simple pharmacokinetic model, and he includes background information about this, as well as allows the user to customize some of the options with regard to the parameters in there. And so it's a very nice tool for people who are concerned and have these levels measured to see what their serum levels would be over time if they were to cease exposure or to have a new exposure. And what I want to conclude is just show how this calculator could be used with some of the epidemiological data we have. And I'm going to use some data from a paper that was done by a former postdoc of mine, Megan Romano, who's now at Dartmouth College, where she looked at the relationship between PFOA exposure and early termination of breastfeeding. Specifically here, I'm showing the relative risk of quitting breastfeeding at 3 months in the second column here. And that's as a function of the woman's PFOA concentrations during pregnancy. And I've cut this up into quartiles, so the first, second, third, and fourth, with the first being the reference group. And so we saw statistically significantly increased risk of quitting breastfeeding early with increasing PFOA exposure. So it suggested that PFOA was doing something to decrease breastfeeding duration. What I did here in this slide is I assumed a background serum PFOA level of 2 nanograms per milliliter, and then I used Scott Bartel's calculator to input various water serum PFOA levels to then create the serum PFOA level we observed within each quartile of the study that Megan did. And so the water PFOA concentrations there in the third column are the levels I inputted, and then that's the serum PFOA level in the fourth column that would result from both background contamination as well as that drinking water contamination level. And what you see is that at levels at or below the current EPA health advisory limit for PFOA, there are increases in the risk of early termination of breastfeeding that are significant in our particular study. So this suggests that these health standards that have been promulgated are not necessarily completely protective of this particular endpoint. Now, there's assumptions that went into making this calculation and as well as the study that I'm showing you this data from, but this is meant to be provocative to make us think about whether or not that this is sufficient to protect human health. So to conclude, I've presented some evidence here to show that PFAS exposures may have adverse health effects on the developing fetus and infant and child, specifically with regard to fetal and childhood growth, breastfeeding duration. And there are ongoing molecular epidemiologic studies that are trying to elucidate mechanisms that perfluoroalkyl substances may affect infant and child health. Also, that the current water-based drinking water health advisory standards may not be sufficient to protect public health, and we may need to be doing more to ensure that there's adequate protection. Thank you.
All right, thank you, Joe. This is both really interesting talks. Thank you both very much. So now we are open for questions and answers. And if you want to submit a question, please go, I believe, to the top of your screen. There's a Q&A button, and you can just click on that and type in a question. And then I will read them. And I do not see any yet, so I will start. I'm going to go back to Allan first. Allan, you mentioned really briefly that sometimes you see the same epigenetic markers from the same exposure in like babies and adults. And I'm just wondering if you can elaborate on that a little. Like, is it, and so why would the early exposures be more harmful? Because they last longer, or are they different? Are the epigenetic changes the same, but then they just last longer and they're more important because you're developing?
Well, yeah, so let me clarify what I meant by that. So I think that sometimes when we're talking about a biomarker, we're talking about a biomarker of exposure. So some sort of measurement of internal exposure. And so I was saying that babies that are born to women who smoked, they have some of the same epigenetic marks as adults who used to smoke. I think that that's kind of a remarkable fact, because we think of babies as sort of starting fresh, right? And yet, you know, there's already this indelible signature of their at least fetal exposure. And other people are working on trying to understand potential transgenerational mechanisms, although that work is very difficult, particularly in humans. So I think that we know many biologic mechanisms through which children are more susceptible, but I think it's kind of remarkable that we can trace these exposures and methylation signatures back to exposures that happened prenatally. We know that they're even stable into childhood. We see that. Joe mentioned in his study they're going to be looking to see how persistent are these signatures of exposure. I think that's really important work. And we know from studies, you know, the example I gave was with adult smokers. You can tell that someone used to smoke for years after they've stopped, right? And so that fact alone is of interest to groups like insurance companies. They want to use this to risk stratify people, which is a little bit scary. But your epigenome is something of a passport, which has the history of all the places that you've been at some level, right? So that's what I meant. Not to say that it's sort of worse for adults versus kids or vice versa, but this commonality of the biologic mechanism potentially, and that we see the same signatures, that also makes us feel that this is more likely to be something quite robust, right? That this is turning up in multiple studies, the same associations, and even across, comparing sort of former adult smokers and babies born to women who smoked.
And then there's one question about this, sort of, I can just tag on to that. What about babies of dads who smoke?
Yeah, so the importance of the dad is something that's under study right now. So Rick Pilsner at UMass has some studies going on paternal exposures and impacts on. He's looking at the epigenome of sperm using some of these same high throughput tools, the microarrays that I mentioned. So I think much more needs to be understood, and that's definitely an emerging area. Yeah. Really interesting.
And I'll add to that, there's been work that has been done in this area in the animal literature looking at paternal exposures from Olivier Rondeau's group, as well as Tracy Bale's group, Diaz and Ressler, looking at paternal stress or paternal preconception stress and showing that there is some potential for the dad to pass on some of this information via the sperm to the offspring. And Dr. Russ Hauser and I of Harvard School of Public Health just recently are going to initiate a new study to follow up children from a preconception cohort and look at how environmental exposures may affect children's health, with the goal eventually being to understand some of the biological mechanisms. But I think this is a really important area for us to be studying. And in terms of human studies, there have been a couple of them done with regard to phthalate exposures by Carmen Masurlian from Harvard School of Public Health, looking at the relationship between phthalates and birth weight, as well as phthalates and neurodevelopment in children.
All right, thanks. So, okay, now we're going to start at the top. What's the proposed biologic mechanism between PFOA serum concentrations in mothers and early termination of breastfeeding? So, unmute.
Yeah, so this is an interesting question. It's not, we don't have a really good understanding of the mechanism in the epidemiological studies. There's some evidence from the animal studies that these could influence lactational hormones like prolactin, as well as breast development. It's important to remember that for women, breast development doesn't finish until they've breastfed. That's actually the last stage of breast development in sort of natural breast development, because there's changes in the tissue that result in the production of milk that are necessary. And so it's possible for chemical exposures to have this sort of developmental effect at a later stage of life when this tissue is still about to go through its last developmental stage. And I think this is a really complex relationship because breastfeeding is really a dance between the infant and the mom, and they're a dyad. And with these persistent pollutants that are found in breast milk, the infant's getting perfluoral substances while they're feeding, which may affect the taste and composition of the milk. The perfluoral substances may affect how the infant responds to feeding, or affect how their feeding behaviors. The prenatal exposures could have affected them. They could affect the mom. They could affect how the mom delivers that milk and interacts with the infant. So it's really difficult to untangle this mechanism, but it's remarkable that it's so consistent across 3 studies.
All right, thanks. Joe, continuing, there's 2 questions about sources of exposure to perfluorinated compounds. One mentioning Teflon is getting a lot of press. Are there other major sources we should be worried about, and like what in our diet increases exposure to these compounds?
So I don't think we have a real good sense of what specific dietary components are contributing. The literature is sort of all over the place there. And I think the safest thing to do from a public health standpoint is to hedge and say you should eat a balanced diet, and try to spread the risk around as much as you can. In terms of other substances, as I mentioned, there's thousands of these, and even among the ones we know to look for, we only know a few dozen of them. And so, you know, this is really what we're studying is really the tip of the iceberg with these. And so with regard to other routes of exposure, you know, I think some of this I was maybe a little overly simplistic. I think in some cases, as I mentioned, with certain communities, water is important, but then in other cases, for young children, dust-borne exposures from hand-to-mouth activity could be quite important, particularly in environments where there's lots of dust-borne perfluoros. So there is some developmental timing to this as well.
All right, Allan, what do the cells in the cord blood represent? Like, are those baby exposures, you know, specifically? And if so, if it's the baby, like what cells, and how do we know, like, do we know if that relates to what's going on in the baby tissues? The baby's
Sure.
tissue.
Yeah, so the cord blood it's the baby's blood, but it's a sample that we're able to get, you know, at the time of delivery. So the cord blood, the DNA in the cord blood is made up of a mix of different kinds of white blood cells. So, you know, the cells of the immune system. And so when we think about what kinds of health effects might those be relevant to, we know that, you know, white blood cells play a role in, you know, immune responses, sort of the defense mechanisms of the body. We know that they're important for inflammation, but that's a tissue that's, you know, a little bit far afield from, for example, what's happening in the brain, right, which has this blood-brain barrier. So the cord blood may not be a good proxy of what's happening in the brain. We did some work trying to use statistical methods, machine learning, to see whether you could sort of extrapolate. Could you understand what was happening in the placenta, which is another tissue that's you know, exquisitely important around that time and plays a key role in development, but is very different than the blood? Could you try to look at the methylation of the blood as a proxy for what's happening in the placenta? And we found that by and large you can't. So I think that, you know, tissues are, as I mentioned, they have their own epigenetic patterns, and they may have their own epigenetic responsiveness. So, you know, in thinking about the kinds of studies that you want to undertake, depending on the health outcomes that you're interested in, or depending on the mode of action of the kinds of exposures that you're studying, you need to get a little bit creative about what sort of samples to collect. So blood is very commonly measured, but, you know, earlier we talked about sperm. If you're interested in paternal impacts and paternal associations, other people do, you know, they do swabs of epithelial cells from the nasal passages if they're interested in what's happening in the upper airways. You know, you usually can't get a biopsy of someone's lung. So people have been getting creative about what sorts of biologic specimens they can get, but that's definitely a challenge in these health studies, because there's only so many tissues that people are willing to part with.
Right. All right, Joe, is there any direct or indirect evidence that the molecular mechanisms that lead to lipid accumulations of PFAS in the mouse liver also would apply to humans?
So perfluoroalkyl substances don't typically accumulate in lipid-rich tissues. They're persistent pollutants, but they're not. We typically think that they're highly protein bound in the serum, and then that they go into interstitial spaces, or the space between cells. Can you say the question?
But I think he means that lead to lipid accumulation.
Oh, I see. Yeah. So yeah. Yeah, I think the species differences are hard to understand. I don't have a good answer. And I was just talking to some toxicology colleagues here at a meeting I'm at, and you know, mice and rats metabolize cholesterol very differently than humans. So it's possible that it could just be that there's different mechanisms going on, and that the perfluoro substances affect these different mechanisms that might not be as important in animals or in humans, but are important in animals, and vice versa. So we don't have a good answer on that, you know, and
okay.
It's surprising though to see this, given that in mice and rats, the half-life of perfluoroalkyl substances is on the order of hours to days, whereas in humans, it's on the order of years. So there's differences just in the way we excrete these substances in these two species. Monkeys are different too. So if you have just differences in excretion and half-lives, you know, you could have differences on other mechanisms as well.
Okay. All right, we still have like 5 more questions, which I think we'd have time if everybody could stay for a few more minutes. Our speakers have said that they can. So I'm just going to keep going if that's all right. Let's see. What about sample size to study the whole genome? Alan?
Sure. Yeah. Well, in studying the epigenome, right? If you're thinking about these methods, that Joe was talking about, which he's undertaking, with these microarrays that I was presenting about, it's a challenge. And if you want to be able to hone in on the specific signatures and be able to sort of exclude chance, yeah, we're finding that this is a difficult undertaking if you have a smaller study, particularly because the associations that we see, even when they're very, very consistent, they tend to be pretty small in magnitude. So the difference in methylation between in the children of the women who smoked throughout pregnancy versus the women who didn't smoke throughout pregnancy, even though we see it in every single study, we're talking about a few percent difference in the methylation in those top sites that are most strongly associated. And so when we do power calculations, I mean, yeah, I think it's useful to have hundreds of samples. Obviously, you have more power, you have more statistical ability to find these associations the bigger your data set, but that can be made up for if you have a really strong exposure contrast, if the variation in exposure is quite large, or if you use some of those strategies that I was promoting where you kind of focus or target what you're looking for, prioritizing things that you've measured well, or where you think that you might be more likely to see those associations because of their biologic context, even in a hypothesis-free way. We know that certain contexts arise more often and that not all sites are equally good candidates. So I don't want to discourage people if they have a smaller study, but yeah, there are some barriers to entry, and these arrays, while very efficient, they are still kind of expensive as an undertaking. They're several $100s each, and so doing several hundred samples at several $100s each is a large undertaking.
All right. Thank you. We got a thank you. Great talks. How persistent over time may be the associations seen with DNA methylation, for example, in the case of PFOA and the DNA methylation at specific genes? We have good evidence. And it sounded like, Joe, you were researching that. I don't know if you've done it yet. Or...
Yeah, that's the next step. We're just finishing up a new follow-up on children in our cohort at age 12 years, where we're going to collect blood and then do those DNA methylation assays in the blood from 12 years, as well as the cord blood, and then we're hoping to also do it for at least one other time point. That's an open-ended question. We don't know the answer to that, and Alan can probably answer it better for some of these other exposures. But as you mentioned, tobacco smoke, we see those signatures for quite a long time even after the exposure has ceased.
Yeah, and I would just add also, it depends a little bit on the tissue that you're looking at. So, some kinds of cells turn over much more rapidly, and so maybe, that they're going to have less of that epigenetic memory, if you will. But I think that the answer could vary depending on the exposure. And so it's important to undertake the kind of research that Joe mentioned.
And it's important to note, and Alan and I have talked about this before, that in some cases the changes we see may actually be adaptive to an exposure, right? So if you're looking at, you get a cold virus, you want your immune cell profile to change to adapt to being able to fight that infection, and so you need those changes. And so it's possible even with chemical exposures there could be adaptive changes to help metabolize them or excrete them, and they're not necessarily of a adverse health effect consequence, but they're actually adaptive in response to the exposure.
All right, next question. Joe, how do you address potential confounding of nutrition and diet that affect both PFOA levels and also obesity?
So in the studies I showed you there, we've adjusted for lots of potential confounding factors. Diet is a really important one that, arguably a lot of environmental epi studies have not done a really good job addressing with regard to diet-based pollutants. It's difficult to assess diet well in pregnant women and children. And so in the newer studies we're doing, we are assessing diet using some gold standard like measures, using things like 24-hour food recalls. And And that's something we're gonna adjust for in future analyses, as well as physical activity. But one of the things that's sort of interesting about the perfluoroalkyl substances is that the concentrations don't vary substantially by a lot of socioeconomic factors. They vary, but not in a way where, you know, poor women or poor people or minorities have higher levels. In fact, it looks more like that people that are more affluent and white tend to have higher levels of some of these perfluoroalkyl substances than those that are poor or minority. So in that case, because those factors are associated with better health, we may actually have negative confounding in some of these cases where our associations get stronger after we adjust away for those factors.
All right, real quick, any research on health outcomes related to prenatal exposures to FTOH, precursor to perfluoroalkyl substances?
No, not that I'm aware of. There's interest in it, and there's a lot, you know, both at the regulatory level and you know, in scientifically. But you know, these are the fluorotelomere alcohols that are used as precursors to some of these and used in the to make the polymers. And I'm not aware of any. And you know, we don't really have good biomarkers of them either. From what I understand, they go in and out pretty quickly out of our body, and in the environment they're degraded pretty quickly. But there is exposure from some food packaging, from what I understand. And then, you know, endogenously and exogenously, they can be broken down into the carboxylates. And so eventually all fluorotelomere alcohols turn into a carboxylate.
All right, Alan, are people doing controlled exposure studies in animal models and looking at the epigenomic effects as another way to validate or add confidence to epidemiological findings?
Absolutely, right. So I didn't speak about it, but there's a, you know, often epidemiologic universes have a parallel world in which people are doing controlled experiments. We have to use slightly different tools when we're looking at other species because you know, the sort of the street address of an epigenetic mark has to do with where it is in your DNA sequence. And there's some similarity in sequence, but you know, you can't use the same exact chip that we use for humans on other more dissimilar species. But yeah, there's a lot of evidence and a lot of people have done some amazing work. There's some prominent work from Michael Meany's group about licking and grooming and how sort of early rearing of pups, of small rodent offspring can have an impact on their stress response, and that's seen through their epigenetic marks. There was some classic work on how BPA exposure in the viable agouti mouse model can alter their both coat color, which is a very physically obvious phenotype, but also their likelihood to be obese. So there are epigenetic models. Again, sort of like Joe's point earlier, sometimes it's hard to know how that translates back to humans. In the case of the viable agouti mouse, although that work is very well established, it's kind of a weird genetic quirk that makes it happen in that mouse. And so it's not really, it's a proof of concept, but it's hard to see exactly how that's operating in the humans. So I think we're always in environmental health, we always have this sort of back and forth where we can interrogate and ask different kinds of questions in the experimental setups versus in the observational, more epidemiologic approaches.
All right, thank you. And we are totally out of time. We're 10 minutes late, so I'm going to turn it over to Maria.
Great, thank you so much, Sarah. We are approaching the end of our webinar today. A video recording will be available on the CHE website soon, and tomorrow you will receive an email containing a link to the video. CHE's next call from the CHE Alaska Partnership will take place Wednesday, February 28th, and will feature investigative reporter Sharon Lerner. Details will be available on CHE's website soon. CHE's next webinar in this series, 20 Pioneers Under 40 in Environmental Public Health, will take place on Thursday, March 1st, and is titled Environmental Chemicals and Preterm Birth, Emerging Threats and Priorities for Future Research. To learn more and to RSVP, please visit our website at healthandenvironment.org. If you are new to CHE and would like to stay updated about upcoming events and more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website. Additionally, if you appreciate these CHE Partnership webinars bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers once more for taking the time to present today, and Sarah for her excellent moderation. Thank you so much for joining us, and have a great day.